This is Bio 181, Chapter 14 on DNA. This is a general overview of a few characteristics of DNA replication. So when DNA, let's just back up here. So when DNA makes a copy of itself, right? So it copies, that makes more DNA. This process is called replication. And if you remember, in a eukaryotic organism, replication occurs in interphase in S phase, where DNA is synthesized. Okay. And if we were to look at the chromosome, it starts off as a linear chromosome in a eukaryotic organism. And then we learned that an exact copy of this was hooked onto it and connected to it with a centromere. And we had two sister chromatids, right? So we copied the DNA, so we had the same sequence of genes, exactly the same alleles in this case for the sister chromatids. So now we're going to be talking a little bit more about this process, okay? Not just when it happened. So semiconservative replication, DNA has two strands. So we've got, I'm just going to represent this as like the sugar phosphate backbone here. So a sequence of nucleotides, we've got one strand on the top, you've got another strand on the bottom, sequence of nucleotides. One strand is going what we call the 3' to 5' end in this direction. The other is anti-parallel, which starts at the 5' end and ends at the 3' end. They are connected with nitrogenous bases. So we could make it a little smaller. Okay. Right. So if I have A here, it's going to be, I'll just come up with a strand here. So I'm just going to randomly think of some sequences. Okay, those are the bases. So these are some bases on my first strand. And so the other strand will have complementary pairing. So with A, T is going to be paired. With C, G is going to be paired. Remember, cars in the garage and apples in the trees. So garage, car, apple, tree, apple, right? And they are connected with those hydrogen bonds, right? So the bonds between them, 2 hydrogen bonds between A and T, 3 sets of hydrogen bonds between C and G, okay? So when we go to make a new DNA strand, all we have to do is break this apart, okay? So cut these hydrogen bonds, right? Cut or break the hydrogen bonds, and you use each of these to make a new strand. So you start with what's now the template strand, which I'll represent with red. That's our original one in both cases, okay, bottom and top. And we would just use the sequence. I won't do the whole thing, but if on this side up here, I have T, G, C. On the bottom, we had A, C, G. Okay. Then you would use that if you make the new strand. Let's say the newly synthesized strand is going to be represented in blue here. Then all the enzyme has to do is recognize that A is present, and it's going to add a nucleotide T. It recognizes that C is present, and it's going to add a nucleotide with G. So this just happens over and over again. Anytime DNA is replicated, we're going to split apart the hydrogen bonds, and then we can just use that to make a complementary strand. The original strand that's used is called the template strand. So this is the template strand. And it's semi-conservative because half of it is old. So this is going to be old, right? And then half of it is new. So half of the molecule is going to be the original and half of it is newly synthesized. So hence semi-conservative replication. Here's just showing this template strand in a diagram from your book. So our template strand over here, okay, sugar phosphate backbone. Again, each of these is our nucleotide monomer that is connected to the next. Okay. Repeating units. Some of them are A's, some of them are T's, some of them are G's, some of them are C's, but they're all nucleotides. They're all connected with hydrogen bonds to the new strand. Okay. And so again, when we're down here at the A, at the base of the template strand, what happens is an enzyme comes along and it fits. The only thing that's going to fit and catalyze the reaction with A is T. So with adenine, always thymine is going to be bonded, and then it moves down, the enzyme will move down to the next nucleotide. Prokaryotic replication has one origin and replicates circular chromosomes. So if you remember, here's a prokaryotic cell. A prokaryotic cell does not have a nucleus. So the DNA in the prokaryotic cell is just hanging out there in the cytoplasm, right? So here's my DNA. And it's actually a circular chromosome. So when it replicates, there's going to be one area where it starts. That's called the origin. And then it's going to open up at this place, and it's going to create two strands. I'm just going to show the DNA here. Again, we're going to be using the template strand. Whoops, gosh. Right, so in this case, the blue is my template strand. I'm using that if I'm the enzyme creating DNA to make the complementary strand. Each of those are going to be replicated and then ultimately passed down to two new prokaryotic cells. Okay. And that's in contrast to eukaryotic replication. So we know that in eukaryotic cells, we have a nucleus. Inside of the nucleus, there are multiple linear chromosomes, right? We have 46 of these in our human cells, right? Our somatic body cells. So we have lots of these. And every time that the cell is replicated, we are going to make a copy of these. So if I just take one of these DNA strands, it's going to be replicated, again, semi-conservative replication. So the template strand is going to be broken apart. The hydrogen bonds will be broken, and then it's used to create the daughter strands here. And as we know, in the case of S phase or interphase, these would actually be connected with centromere. So it would look like, instead of separate like this, oh no, it would be a little bit closer. Oh no, and it would be connected with the centromere here in the center. So this is supposed to be kind of like an X. There we go, right? It would look more like this. Just trying to tie it into what we learned previously with the sister chromatids. All right. That's it for this video.